Hello, this is Jim Schnabel with the 23rd lecture in a series from Principles of Biochemistry, 7th edition, Albert Lehninger author. Today we'll be beginning with talks about regulation of these glucose and gluconeogenic pathways. We'll begin with discussion of the pentose phosphate pathway that you will see in rapidly dividing cells, including those with neoplastic transformation. Primary products in these systems are NADPH, which is used for protection against free radicals, and 5 carbon sugars for nucleic acid. So these are the means by which we generate the ribose moiety for amino acid or for nucleic acid production. The pentose phosphate reactions are shown here. We begin with glucose 6-phosphate. Up at this point, there's a glucose 6-phosphate dehydrogenase with production of NADPH, and from this you get a 6-phosphoglucano delta lactone here, which is the operative oxidation of this carbon right here. The next step is a lactonase, where water breaks the bond, and you get 6-phosphogluconate from this. So the terminal, you have a carboxyl group at this carbon right here, and the presence of a hydroxyl group that cuts in and attaches at this point right here. The next step is 6-phosphogluconate dehydrogenase, which produces an oxidation of the 6-phosphogluconate with production of NADPH and liberation of carbon dioxide. So that works off of this entity here, and this is the part of the molecule that leaves, and you get oxidation at this point. This is generally the situation where you get a redox reaction here, oxidation at this point with liberation, and then you pick up the proton from solution. And then after you get from that you get the 5-carbon unit, D-ribulose 5-phosphate. Then there is an isomerase reaction, and with that you get ribose 5-phosphate with involving an oxidation at the carbon terminus right here, and a reduction of the number 2 carbonyl right here. So we get this structure, and this is a structure that we employ with nucleic acid backbone production. Additionally, you can recycle the pentose phosphates to glucose 6-phosphate, and that is partitioned between glycolysis and pentose phosphate pathway. Among principles of metabolic control, these pathways under discussion are interconnected and regulated together. The term homeostasis implies a steady concentration, the amount of substance, and its associated catalytic enzyme activity are regulated. This is an example of that. We have in this schematic extracellular signals, which are recognized by receptor. Transcription factors are added. We use a DNA template. We get messenger RNA, which can, of course, be degraded or be translated, producing enzymes, and these enzymes have a variety of different fates. They can either be involved in protein degradation, they can be sequestered subcellular organelles for production. You can have it do its usual role of binding substrate and reactions, binding ligands. It can be involved in phosphorylation or dephosphorylation reactions, and you can combine it with regulatory proteins and get allosteric and regulatory effects that way. In this example, we have adenine nucleotides also providing regulatory roles. These are inhibitory steps here with AMP kinase. You can have stimulation of this entity through exercise, through increases of AMP or decreases of ATP. So there is an energy requirement here. Sympathetic nervous system can also stimulate this via signals from the brain. You can have effects on food intake, heart, and utilization of energy with decreases of insulin secretion, fatty acid synthesis, and lipolysis. So these are energy-inducing effects in this particular schematic here. AMP, activated protein kinase. The rate-limiting step is not limited to a single enzyme. It is instead several enzymes are involved, and each enzyme contribution to control can be measured individually. In a lab experiment, you can add enzyme to the excess and determine resulting activity. The flux control coefficient is a relative amount of enzyme which contributes to the pathway, and these can be measured for each enzyme in the pathway. These coefficients for all the enzymes is equivalent to 1, and the response coefficient is where you can mathematically quantify the effect of a cofactor, hormone, or growth factor on a pathway. We talk about carbohydrate metabolic control. Hexokinase is more important than PFK1 in glycolysis regulation. Gluconeogenesis depends on fine control phosphorylation, and to increase flux through a pathway, you increase the amount of enzymes in that pathway. And gluconeogenesis control is governed by the amount of ATP. Both of the competing glycogenic and gluconeogenic reactions utilize ATP, and this is what we call a feudal cycle. Again, as a point of review, we have glycolysis with pathway enzymes in common here, here, and here, and then we have the three enzymes which are out of phase, the pyruvate carboxylase, PEP carboxykinase, and the glucose 6-phosphatase. All the other ones are in common with this. Here's the fructose 1,6-diphosphatase. So I guess there are 4 right here. Isozymes are different enzymes which catalyze the same reaction. They are given the same name, but they're location specific. There are, for example, 4 hexokinases. They have different locations of action and kinetic behavior and differences in regulatory behavior. Fructokinase 1 and fructose 1,6-diphosphatase are also reciprocally regulated. ATP inhibits PFK1. It binds to the enzyme and lowers affinity for the substrate fructose 6-phosphate. AMP inhibits the reciprocal enzyme in gluconeogenesis. So they are opposing actions here, ATP inhibiting PFK and AMP inhibiting the phosphatase enzyme, which is involved in gluconeogenesis. The PFK product, fructose 1-bisphosphate, inhibits PFK1 and fructose 1,6-biphosphatase, so it's direct feedback inhibition. The enzyme xylose 5-phosphate, which is a product of the pentose phosphate pathway, mediates glycolysis, stimulates glycolysis, and inhibits gluconeogenesis. Pyruvate kinase exists as 3 isozymes. The glycolytic enzyme is inhibited by ATP. The fate of pyruvate, there is conversion under strict control. It can be converted to acetyl-CoA or oxaloacetate. Acetyl-CoA inhibits pyruvate kinase and activates pyruvate carboxylase, and that is the first enzyme that is involved in gluconeogenesis. and stimulates that pathway. Transcriptional regulation, insulin regulates about 150 genes, is involved in transcription regulation of messenger RNA. The endpoint is glucose consumption and inhibition of the production of release of glucose into the blood. We talk about carbohydrate response, element binding protein. These coordinate carbohydrate and fat synthesis. The dephosphorylation allows for transcription factor to enter the nucleus. So again, this is representation of the same series that we've had before between glycolysis and gluconeogenesis, the fine control that exists over the particular steps in here. There are other transcription factors. There's a sterol regulatory element binding protein called the SREBR1C, and cyclic AMP response element binding protein, which is CREB. For glycogen metabolism in animals, glycogen is about 10% of the liver weight, 1 to 2% muscle, and they serve respectively as a storage depot in the liver and a source of rapid energy in the muscle. Glycogen granules can store glycogen and degrading enzymes. Glycogen phosphorylase removes the glucose terminus and also utilizes debranching enzymes. So storage form has this as the initial glycogen phosphorylase to remove the terminal glucose. Glucose 1-phosphate product is converted to glucose 6-phosphate by phosphoglucomutase. It enters the glycolytic pathway. Glucose from the liver is then released into blood. So the glycogen synthesis substrate is UDP glucose. Is glucose attached, number 1 carbon, the UDP, and it is synthesized by UDP glucose phosphorylase. And this is an example where you get sugar and then the particular NTP. This can be any nucleotide, adenine, guanine, and you have the sugar attached with a bipyrophosphate created with the sugar on one end, ribose on the other end. This is a sugar nucleotide, NDP sugar, and you get inorganic pyrophosphatase causing 2 phosphate ions, and this is the net reaction, sugar phosphate plus the NTP giving the NDP sugar plus free phosphate. There's an immediate donation of glucose to the glycogen chain. To branch the glycogen chain, you use the primer molecule glycogenin that acts both as a chemical primer and enzyme to the branch chain 1,6 linkage. The glycogen regulation, glycogen phosphorylase A is active, glycogen phosphorylase B is inactive, is converted to the A form after epinephrine or glucagon stimulation. Cyclic AMP is the second messenger. The B form of phosphorylase is phosphorylated to produce the A form. Low glucose triggers the release. Phosphorylase B kinase uses cyclic AMP cofactor, and this is opposed by a phosphoprotein phosphatase. This progression is a cascade. For glycogen synthase, we talk about the phosphorylases form in the active A form, and the active A form is dephosphorylated. Glycogen synthase kinase 3 is the most important of the glycogen kinases. It is heavily regulated. It mediates insulin activity, which inactivates glycogen synthase 3, and this increases glycogen synthesis. This schematic here with insulin, attached to insulin receptor, you get conformational change. Then the IRS1 is produced. You have protein kinases that are invoked here, and you have activation with the dephosphorylated form, and glycogen synthase is produced again with dephosphorylation to the active form, and the pyrophosphatase works at this point with liberation of phosphate. All this is triggered by the binding of insulin with these various cofactors that come into play. So pyrophosphoprotein phosphatase 1 is activated by insulin. It activates glycogen synthesis. There are allosteric and hormonal signals. Well fed is the insulin state. Fasting, you get increases of glucagon. Epinephrine is a fight or flight hormone. And the muscle is a standalone site for glycogen utilization. And that will be it for our talk. Thank you very much.